Hello everyone, welcome to the student presentation week 2025. Myself Gayatri Rajkumar Singh Bais, student of B Pharmacy 5th semester of P.R.Patil Institute of Pharmacy. Today we are going to discuss on the topic of GENTIAN. Presentation on GENTIAN. Contents are synonyms, biological source, geographical source, chemical constituents, collection, morphological and microscopical characters, extraction and isolation method, chemical test, marketed preparation, adulterants, contraindication and adverse effect and uses. Gentian synonyms are yellow gentian, bitterroot, bitterwort, great yellow gentian. Biological source are the specific source is the dried roots and rhizome, underground stems of the permeable herb Gentiana luteosa, which belongs to the Gentianaceae family. Geographical sources are Asia. It is found in Central and Southern Europe. Cultivation Gentiana luteosa requires a moist soil, good range, and a suitable soil consisting of low peat and grit. Chemical constituents are gentian consists the bitter glycoside gentianopicrin 2% as the principal active constituent on hydrolysis. It yields the glycon, mesogentinogenin and glucose. Gentinopicrin is a saccharoid gentiopicroside and it is decomposed on fermentation and drying of the drug. Other bitter compounds are genticin. It is the structure of gentiopicrin is a chemical constituent of gentian. Collection. The plant is large permeable herb. The drug is collected from 2 to 5 years old plant in autumn. September to December, turf is stripped and the rhizomes are dug up. It is cut into pieces of different length and dried quickly, first in air and in shade. Morphology of gentian. Color brownish, shape subcylindrical, entire or longitudinally split pieces of rhizomes and roots. Size 50 to 20 centimeter or more in length and to 5.8 centimeter in thickness at crown. Morphological characters. First, pedidrum, cork, 4 to 5 layers, thin walled, rectangular cells and orange-brown in color, phylogeny and phylodrum, not distinguishable through a few layers of thick-walled, tangentially elongated from parenchyma is seen immediately below cork. Second, cork. Thin zone of tangentially elongated, thick-walled parenchymatous cell containing oil globules, few starch grains and minute acular raphides. Third, secondary phloem. Relatively a wider zone made up of tangentially elongated, thick-walled parenchymatous cell containing oil globules, few starch grains and minute acular raphides. Fourth, cambium. Distinct and represented by yellow ring of 3 to 5 layers of thin-walled, small rectangular cells. Fifth, secondary xylem. Forms the bulk of the root is consist of largely parenchyma, vessels and medullary rays. Microscopical structure of gentian. Extraction methods. First, solvent extraction, maceration, suspend plant material, example leaves, root, flowers in a solvent like ethanol or hydroethanoic mixture, example 70%, 50% hydroethanoic macerated over night for complete extraction of compounds. Reflux extraction. Use heat. Increase extraction efficiency such as replacing with 70% ethanol or 90% ethanol for several hours. Isolation methods of GENTIAN. First, solvent partitioning. Redissolve the dried extract a solvent and perform liquid-liquid extractions with different solvents. Example, chloroform and butanol to separate compounds based on their solubility. Second, chromatography. Column chromatography use silica gel or DAE cellulose columns with different solvent gradients to separate fractions containing different classes of compounds, example flavonoids and polysaccharides. Chemical test of GENTIAN. First, alcoholic extract when made alkaline shows blue or green fluorescence. Take moistened dry powder in test tube, cover test tube with filter paper, soak in dilute NaOH, keep test tube in water bath. After some time, expose filter paper to UV light. It shows yellowish green fluorescence. Marketed preparation. Gentian violet is a solution used as a antifungal for wounds. Adulterants of GENTIAN. Adulteration occurs due to careless collection. The rhizomes of Rumex alpinus give the test for anthraquinone derivatives. It is a dangerous but easily detected admixture with the rhizome of Veratrum album. Contraindication avoided in pregnancy and lactation. Contraindicated in gastric and duodenal ulcer. Adverse effect. Raw extract may cause nausea, vomiting and diarrhea. Uses. Potent stomachi and treats GI problems like indigestion, immunogogy. This bitter stem lace is used to treat wounds, to treat arthritis, sore throat, and jaundice. Acknowledgement. I express my heartfelt gratitude to Dr. Nilesh Banarse sir, our estimate guide, for his continuous support, motivation, and valuable guidance. I am thankful to respected principal sir, director sir, all the faculty members and non-teaching staff for their encouragement and assistance. Special thanks to my friends and classmates for their cooperation and moral support. Thank you.